Good morning, and thank you for tuning to our fifth webinar of our series on environmental DNA. Our topic today is metabarcoding or targeted qPCR for environmental DNA surveys. My name is Mario Thomas, co-founder and CEO of Precision Biomonitoring, and your host today. Joining me for this webinar is Dr. Steve Crookes, Head of Development at Precision Biomonitoring, and Rémi Maglione, co-founder and Metagenomist at AgriSeq Solutions, a strategic partner co-located in Guelph, Ontario. If during the webinar you lose the sound or image, you should refresh your screen to solve the issue. Also, make sure you close all other programs and apps that you may have opened on your device. As we all know by now, environmental DNA or eDNA is a relatively new science that has started to seriously revolutionize industry's approaches to biomonitoring, mainly because of its well-proven accuracy compared to traditional methods. And also the time it can save to conduct biomonitoring surveys and cut costs. There are now over 500 scientific papers published in peer-reviewed journals, proving the advantages of eDNA for many applications. As a result of the strong scientific evidence, adoption of eDNA methods to replace conventional ones is accelerating. In Canada, in 2019 so far, we have seen as much as 4 times greater use of eDNA methods compared to last year. Of course, we are seeing early adopters, more adept to accept the scientific evidence, and it's only a question of time for everyone, including regulatory agencies, to support switching to eDNA for more reliable survey results. In the previous webinars, we have discussed eDNA science and the reliable platform Precision Biomonitoring has developed for targeted qPCR, including triple-log test development, survey design, and survey implementation you can trust. On our website, you can see the details of our platform based on the on-site BioMime sample prep kits that give you room temperature stable DNA extracts, the best way to avoid degradation of DNA before qPCR or metabarcoding analysis. We have also discussed the ecology of eDNA, how it behaves in aquatic systems, and in varying environmental conditions. In addition, we have discussed how a strong understanding of the ecology of eDNA can help progress eDNA science, and why it is a foundation on which to design the most statistically valid eDNA surveys. If you have missed any of the previous webinars, they are now available for viewing on our website. Our next webinar, scheduled for November, will reveal how Precision Biomonitoring is working with machine learning and artificial intelligence to develop software tools that can, for the first time, design surveys that have known statistical accuracy. Our focus today is metabarcoding as a complementary tool to targeted qPCR. Rémi will first cover the basics of metabarcoding, and then Steve will talk about what we believe are some excellent use cases for metabarcoding. Our agenda is a 30-minute presentation, followed by a question period and discussion of 25 minutes. At this point, I will invite Rémi to review for us the basics of metabarcoding. Rémi.
Thank you, Mario, and thank you everyone to being here today with us. So I will present you the overview of the metabarcoding. So metabarcoding techniques are evolving in the world of species identifications. The previous methods mainly called for observations and discriminations of different species based on their shape. However, this kind of approach has several limitations. First of all, certain species can only be identified in their adult form. Early forms of development are not differentiable. On the other hand, the sampling depth is limiting, and it's necessary to have a varying number of resampling, so it's cost and time consuming. With the development of new sequencing technology, we were able to overcome its limitations, especially with the metabarcoding technique. We can define metabarcoding as a technique to directly monitor all your organisms in a given environment. This method requires various skills. In particular, it uses molecular biology with PCR approaches, DNA sequencing, and bioinformatics computation for the analysis of the results. There is a lot of application with the metabarcoding approach. For example, simple species identifications, invasive species detections, food safety, and of course, biomonitoring and ecological assessment. Some of this application will be presented later in this webinar. But first of all, what are the basics of metabarcoding? What is barcoding? So take an example of three organisms, a bird, a mushroom, and a whale. Every of these individual has DNA, and we could use this DNA to identify each of them. So the DNA could be used as a way to barcode each organism. So now we know what barcoding is, but what is metabarcoding? With metabarcoding, we can use the DNA to identify and discriminate multiple targets at once. Let's start with a previous example, but this time with three species of mushrooms instead of one. I know, some of you could argue that for three mushrooms, it could be easy to perform three independent barcoding approach. But think about 50 different mushroom species or 1,000. This is the true power of metabarcoding. So we will follow this particular example for an in-depth visions of the different stages of the metabarcoding techniques. So the first step is to retrieve your samples that contain multiple sources of DNA. The second step is to extract this environmental DNA on site or at the lab. In the third steps, we select mushrooms groups by barcoding with the PCR techniques. Let's take a moment to understand what's going on here. What we do with PCR is to amplify and recover only a selected regions of DNA. Here we have a schematic representations of the double-stranded DNA. The challenge here is to choose a region of this DNA that is common only for mushroom groups. Fortunately, in the majority of cases, you do not have to design yourself, since it exists databases that provide these specific targets. One example is the BOLD databases for barcode of live data systems, which lists multiple target genes, such as the 16S genes for bacteria or the 18S genes for fungi. So you have in your positions, at the end of this stage, the amplified DNA specific to your interest group, here are the mushrooms, and that's what you will sequence. Let's back to our overview. So you're ready for the sequencing. Here is a common, sorry, I had a problem. Okay. Here is a common platform for sequencing that can fit your needs, such as Illumina or Nanopore. I have at least two advices when the time is coming to choose the right platform. So you will need few error rates and a length order of your target fragments. Depending on your font, it will be important to carefully choose the length of your targeted fragment and the sequencing coverage. Why? Because high throughput tech could help us to enrich the relative abundance of each detectable organisms. To make it short, high sequencing depth will be needed to detect rare organisms. After the sequencing, the last steps of metabarcoding is to discriminate your mushrooms thanks to the small differences that exist in their DNA sequences. We perform the steps with bioinformatic tools. Let's take a time again to understand what's going on here. So here is a typical result of sequencing. You have a bunch of sequences with small differences. True biological differences represented by the orange C and black T, and sequencing error in red that are not real. The expected results after bioinformatic processing should regroup the sequencings belonging to each organism. In the past, the OTU approach was used to define groups of sequences belonging to the same organisms. The problem with this technique is its sensitivity to sequencing errors. Here you can see that we have two additional groups of sequences that do not really exist. They are false positives. Thankfully, we now have access to new tools like Day2 that use machine learning to infer sequencing error and get rid of those false positives. The last step is to annotate each group of sequence with a corresponding organism name. This is performed with public or self databases, depending on your project. Let's back to our overview. So you just identified and named all your organisms. One last thing, I do not know if you remember, but our organisms were represented by groups of sequence with a different quantity of sequences within each group. This quantity can be used to reconstruct the original abundance of each fungus in our initial sample. So, at the end of the Metabarcoding approach, we obtain an identification of our organisms of interest and their relative abundance. I think you already understood that Metabarcoding requires lots of bioinformatics skills and resources. At AgriSeq Solutions, we have a strong bioinformatic competence, and that is what we are developing. If you have projects with this kind of needs, we will be pleased to help you. So this concludes my overview of Metabarcoding. I thank you for your attention and return the lead to Mario.
Thank you, Rémi. In a few minutes, you will have the opportunity to ask Rémi questions about this important technology for eDNA. However, before getting there, I'll ask Steve to discuss for what types of questions and surveys Metabarcoding is best suited. Steve.
Thanks, Mario, and good day, everybody. In the following section, we shall discuss how eDNA metabarcoding may be applied in an ecological context to better assist clients in their compliance monitoring of biodiversity. In so doing, we shall briefly touch on the difference between targeted single or few species approaches and large-scale horizon scanning surveys of entire biotic communities, as facilitated by metabarcoding. Following this, we shall discuss the major applications of metabarcoding that have been fully validated, and those applications that promise much for the future. In explaining the utility of metabarcoding, we shall discuss further how eDNA metabarcoding may assist primary and secondary industries and their consulting partners in expediting and maximizing their biomonitoring resources in context, following recent reviews in the academic literature of eDNA as applied to natural resource sectors. Our previous webinars have, by and large, addressed the application of a targeted approach to eDNA biomonitoring. This method, also termed as active detection, ostensibly seeks to determine the presence and/or absence of a single species or a set of few species that are of express interest to the client. Often species at risk or particularly invasive taxa fall into this category, as they are the focal point of any compliance legislation. However, when whole biotic communities need to be assessed, then it is more cost-effective to holistically sample the habitat under scrutiny using metabarcoding, also termed passive or horizon scanning methods. Depending upon the number of barcoding sequences used, one can assess a subset of taxa, for example, purely just amphibians, or even representatives of all life forms found along the entire tree of life from a single sample. The adoption of eDNA metabarcoding to applied ecological issues quickly cemented its role as a hugely informative method to derive massive amounts of information of aquatic and terrestrial taxa from all manner of environmental samples. There are 4 major applications of metabarcoding to the monitoring of biodiversity across various disciplines, spanning presence or absence surveys through ecosystem health. As with targeted surveys, metabarcoding can be used to determine the presence or absence of a particular taxon or taxa. Unique to metabarcoding, though, entire biotic communities can be assessed to determine baseline levels of biodiversity. Thirdly, a subset of taxa, which we call Canary taxa, may be the focus of a metagenomic scan of a naked OTU or ASV data themselves be used to provide a robust analysis of environmental stresses across time and space. Finally, using metabarcoding, one may also begin to identify distinct genetic lineages and populations within species, which may inform upon the decision to confer designatable management unit status to populations. Determining the distribution of species or their absence from a particular ecosystem is the bedrock for much of the species at risk monitoring regulations. The objectives of species at risk monitoring, or indeed other bellwether taxa, may be better served if multiple species can be determined to occupy a site of interest simultaneously. With metabarcoding, presence or absence data can be generated for myriad species from across a broad array of taxonomic categories and all potentially from a single sample. Thus, the costs associated with targeted species monitoring may be more efficiently repackaged if multiple taxa be screened in a single surveillance program. Making this data available can help drive policy directions at the regulatory level, so metabarcoding data may expedite the knowledge flow from the field to the policy desk, and thus aid in the formulation of biomonitoring regulations. Canada's system of lakes provides a great example of the utility of the metabarcoding approach to establishing baselines. Canada is home to many previously unexplored and unnamed lakes that have been subject to no biological evaluation or classification in its post-colonial or indigenous history. A common request from stakeholders, including primary resource developers and First Nations groups that oversee these lakes, is to determine their baseline levels of biota, most commonly for freshwater fishes. In so doing, not only are baseline estimates of fish fauna established, but that also the contribution of indigenous knowledge to biomonitoring is further validated. It has recently been established, for example, that First Nations groups are exemplary stewards of Canadian biodiversity. In PBI's recent history, we have conducted eDNA metabarcoding in partnership with consultant, primary industry, and First Nations stakeholders on remote lakes in both northern Alberta and Ontario, and established baselines of the species that are present. In the previous example, we communicated how metabarcoding can establish a baseline of community composition and species richness, even in systems in which we had no prior or no confirmed evidence of species presence. These baselines established in remote systems represent an optimal case, often conducted prior to the establishment of local primary resource activities, for example, forestry or mining. By employing multiple markers, entire biotic communities, from bacteria to metazoans, can be simultaneously described and the relative changes in their abundance over time and space be documented. Subsequent surveys, each representing a snapshot in time, can track the relative changes in species composition and richness in the event of any disturbance to the environment. These chronosequences would provide the raw material upon which time series analyses of environmental stresses could be conducted. In the world of hydrology and forestry, for example, which include many lotic and lentic water bodies, interested parties could track the temporal influence of anthropogenic waterway features upon aquatic biota, including the impact of dam impoundments, industrial outflows and intakes, channel modification, and watershed land use alterations, for example, urbanization, deforestation, and agricultural land conversion. Mine sites provide, perhaps, the apotheosis of such an approach. The extraction of natural resources through the mining and processing of ores is an economic touchstone of many countries worldwide, including Canada. The extraction of mineral ores, whilst a massive contributor to global economies, leaves a concomitantly huge ecological footprint. As such, there is often stringent regulatory oversight on ecological restoration post-operations. Applied ecologists have recently argued for a holistic approach to monitoring, hence the suitability of metabarcoding to this approach, with the potential to collectively monitor all phyletic levels of life. The higher inclusivity of metabarcoding methods lends towards their use to determine the trajectory of biota once mines have ceased operation and recovery efforts enacted. The metabarcoding of various repositories of eDNA would provide a site reference with which future metabarcoding results could be compared. A multi-barcode analysis of prokaryotic, protist, fungal, and invertebrate diversity would provide a huge amount of biotic information from which to draw inferences as to how badly a habitat has suffered and or how well it is recovering post-restoration. Metabarcoding monitoring may need not be restricted to after the working life of the mine is over. Some mines make significant landscape alterations during their working operations and immediately have to provide biodiversity mitigation or enhancement of habitat in the interim. One example is the rerouting of waterways to make way for an extraction pit. All reroutes are subject to enhancement rather than restoration per se, and eDNA monitoring of multiple aquatic species may be routinely monitored for their response to changes in the system and whether the enhancement has resulted in an increase in abundance and species richness as would be the desired outcome. Primary resource industrial activities produce waste that can injuriously impact aquatic and terrestrial systems. Some organisms are more sensitive to these disturbances than others. Such taxa may represent Canary taxa, an early warning group of organisms whose change in abundance and diversity may reveal the initial stages of biodiversity collapse in the wake of contaminants entering the habitat. Thus, these should be monitored regularly during the working life of a mine, for example, to rapidly identify a pollution event. If a signal of change is indicative of biodiversity loss or change, rapid response action plans may be enacted to remedy the cause and mitigate against significant habitat and biotic loss before irreversible damage impacts the remaining biota. In due course, monitoring using technology such as the MinION may facilitate real-time monitoring of these Canary taxa and be a staple of a biomonitor's toolkit on each and every site of natural resource exploitation. Further opportunities for spatio-temporal analysis of work sites could analyze the numbers of OTUs or ASVs themselves without the need to attach species-specific ID to each. In this way, by merely counting the number and breadth of ASVs, itself represents a metric of biodiversity that prima facie may allow a quantitative analysis of the impact of mining or restoration efforts in maintaining or enhancing biodiversity. As an example, clients could assess quantitatively the impact of a particular activity, be it negative or positive, on the local biota. Time series analyses of species diversity would provide robust data on the nature of the trends in any change in biota as a whole. Metabarcoding can help to identify designatable management units by uncovering the standing genetic variation within a species across time and space. Genetic distinctness plays a role in the assignment of management unit status. According to the Species at Risk Act at the federal level in Canada, some designatable management units have different conservation statuses and are thus subject to variance in the extent of compliance monitoring. Metabarcoding can identify individual barcode variations inherent within species and whether these segregate by putative population or by geographical feature, for example, a watershed or lake. Whilst it is possible theoretically to target individual genetically diagnostic and distinct populations with targeted approaches, metabarcoding of intraspecific variation will provide a much larger and more geographically informative screen of threatened taxa and thus reveal to clients the designation status of their local population or populations of target organisms. The future of metabarcoding is bright as reference sequence databases become more complete, allowing increasingly ever more precise determinations of community composition and species richness. Further applications include the assessment of environmental samples for the presence of pathogen strains in epidemiological surveys and in applications to public health monitoring. Real-time or hugely expedited monitoring would severely reduce the time that expires between initial pathogen detection and the deployment of any mitigation or preventative measures. Metagenomics may also be applied to RNA molecules. the intermediary messenger molecules that communicate the information contained within DNA to the translated protein or final functional RNA molecule. eRNA metabarcoding may allow for the functional characterization of habitats in virgin state compared to when they are under duress and or after a period of restoration. eRNA detects the functional products of genes whose relative levels of expression may change in response to environmental alterations. By tracking this change, one may subsume eRNA detection into our body of Canary markers. eRNA has the potential to be more sensitive than eDNA in this respect, bolstering the sensitivity of routine monitoring of vulnerable ecosystems. Additionally, because RNA is produced by living, viable organisms, we can be more confident that the E-nucleic acid signal represents a local population and has not been transported in from elsewhere or produced by an in situ dead organism. The presence of chronosequences, snapshots of biodiversity in time, may prove to be an essential tool in the long-term monitoring of aquatic and terrestrial systems, and in the monitoring and rehabilitation of sites of resource exploitation and beyond. Although some conventional methods retain the advantage of determining abundance, population structure delimitation, reproductive status, and health of the population at large, metabarcoding approaches may reduce both systemic bias of those methods and reduce the person hours needed to conduct and analyze the data, increasing both the data quality and quantity, whilst also reducing costs. Moreover, third-party analysis of metabarcoding data is highly desirable to the primary resource community, as it adds a layer of accountability to all restoration, mitigation, and enhancement projects. In closing, metabarcoding is an exceptionally powerful tool for data-intensive biodiversity projects whose full potential has yet to be fully realized.
Thank you very much, Steve, for sharing with us your knowledge and insights about metabarcoding and some great examples of precision biomonitoring users for eDNA surveys. I'm glad to confirm that precision biomonitoring is broadening the range of intelligent DNA solutions we offer to best answer your surveillance needs, including metabarcoding and other genomics-based solutions. Should you want to discuss access to our growing technology platform, please contact me directly at this email or telephone number. We now invite you to ask any question you may have for Rémi or Steve, or by clicking on the question button below the presentation and writing them down in the space provided. And we will strive to answer your questions as best as we can. So it's just under the presentation. You see chat, questions, poll. If you click on questions, you'll be able to write down your question. Here's a question from Kanak Bala. Kanak is asking, which sequencing platform are you preferring for metabarcoding? I will direct this question to Rémi.
Yes, so by experience, I mostly used the Illumina platform. So within this platform, you will have to choose the right machine. So usually for small project, I use the MySeq, but for bigger project, you could go for an iSeq or even the new platform, the NovaSeq, that have a huge yield. We are talking about 2.5 terabytes of sequence. No, sorry, not terabyte, tera sequence. Another things to considering is that, so I speak before about the nanopore platform, and I think it's a very promising avenue for on-site sequencing. So, for example, if you have projects that you think it's better to sequence directly on your environment, nanopores should be a solution to consider, because they are really improving their sensitivity and reducing the error rates. Just in the past few months, it was very incredible how they improved the service. And so you will have to have some skills for the nanopore, but it's very easy to use for, I mean, every scientist.
Thank you very much, Rémi, for your insights on this. I have another question here from Soren. Soren is asking how the number of DNA reads captured from different species could be translated to the actual number of those species in the environment, how the normalization is done.
I think that's a Rémi question.
Yeah, it's a Rémi's question. Okay.
So this is a tricky question. So there is a lot of people that are developing tools right now to improve the relationships between the sequence you get at the end of the whole process of metabarcoding and the actual abundance. So for many years, we just use the relative abundance. So it's we use just the number of sequence as a proxy for the true abundance, but it's not the true abundance. And the new tools that are developing are more focused to speak about the absolute abundance. So you raise a good point. So the normalizations is a crucial steps, and the most tricky part of the normalizations is to deal with the zero in your sequences. So, for example, you have a species in a sample that are not represented by any sequence, so you will get like zero count for this sequence, and the whole process of bioinformatics doesn't really like the zero. So you have to choose the good normalizations process, and the last and better normalization, to my view, is the centered zero normalization. So there is a lots of papers and tools that can help you to go with those kind of normalizations.
Thank you, Rémi, for answering Soren's question. So a related question from Ryan that reads, using metabarcoding, what level of resolution do you have when it comes to quantification of detected organisms? In other words, if I see that I have 2x the number of mushroom number 1 as compared to mushroom number 2, is this a real difference?
So it's it's linked to the previous questions. So if you choose the right tools and the right normalizations, you can say that, yes, you can say that if you see more sequence for the mushrooms one, it could be very used as a proxy to say that in your samples you have more mushroom ones. But you have to be very careful of the 2x. It's not because you have 2x in your analysis that you have exactly 2x in your environment. So there is a bit of limitation for these approaches, but as I said before, with the new tools that came in only this year, I have a bunch of paper if you want that I can link for this particular issue. But with the new tools, I think that we are very close to say that 10x 2x for the mushroom one in your sequences will be the proxy representation of 2x on your environment.
Thank you very much, Rémi. Now a question from Scott Taroff that is related to the previous questions, but to bring a more or deeper level. So the question from Scott is, can eRNA and eDNA markers be used simultaneously within the same qPCR reactions to obtain data on, for example, comparing species presence versus functional gene products of those species? Or would the two marker types interfere with each other in qPCR?
I'll field that question. When we're extracting eRNA, we have to extract the RNA and then convert it to DNA before we apply the PCR reaction. So I would probably do the two reactions separately. But RNA is, as you know, is mostly single-stranded for the most part, and is much quicker to degrade as a result. It's more susceptible to endo and exonucleases in the environment. So, but it does reveal like a very recent signal. But because it degrades so quickly, we have to convert it to double-stranded DNA, and we have to have that step. And you could conceivably do that before doing a multiplex PCR. But with multiplex, you do lose slight sensitivity of the assays to the target. So I would probably do the monoplex.
Thanks, Steve, for addressing this. I see that we have covered all the questions that were posted. Are there any other questions at this point in time? So if after this, we close this webinar, you have more questions, you have my email address on the slide above. So don't hesitate to shoot me an email. I'll make sure that with Steve's combined with Rémi's knowledge of metabarcoding that we address your question. If you prefer to have a video call, we can do this as well. So basically, we are at your service to answer any question. And certainly, we have been doing a lot of metabarcoding project this summer. We're doing some more this fall. If you are tempted to do a first metabarcoding study for answering some questions for your clients, reach out and we'll make it happen for you. So I think this will be all for this morning. I thank you very much for spending 45 minutes with us today. You will be receiving in October sometime an invitation to our 6th webinar in November and discuss how to get statistically valid eDNA results. So thank you very much again. I wish you a good rest of the day. Bye-bye.